In this lecture, I will be talking on immunity to schistosomiasis parasite. An infection by this parasite starts by entrance the circuitry into the body, then circuitry transformed into schistosomula, then mature within portal blood vessels and migrate to lungs, then to the liver and to intestinal mesenteric veins, site of infection by Schistosoma mansoni and Schistosoma japonicum, and to veins of urinary bladder, site of infection by Schistosoma hematobium, where schistosomules mature into male and female adult worms. Accordingly, circuitry and young schistosomules may constitute possible targets for vaccine development, which at best limits the parasite load. So, I will just highlight some immune responses to schistosomiasis parasite. The humoral responses to infection with Schistosoma mansoni is characterized by high titers of anti-schistosomes IgE antibodies, which is T helper 2 immune response, results in expression of T helper 2 cytokines, and these cytokines expressed by the T helper 2 are interleukin number 4, which induces class switch to IgE, and interleukin number 5 induces bone marrow precursors to differentiate into eosinophils. Interleukin number 3 stimulates growth of mast cells. And as can be seen here, in infection with this parasite, schistosomiasis parasite, mast cells infiltrate the site of infection, and then they produce granules. These granules, platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor, and this actually increase the infiltration of macrophages and eosinophils. So, also basophils, they take part in immunity to schistosomiasis parasites, as I mean basophils that take part in this immunity to schistosomiasis parasite induce M2 macrophages, arginine 1, that kill helminths by depletion of arginine, and also it secretes proteases as can be seen here, and that recruit more eosinophils to the area of the infection. So these are the recruited eosinophils. They interact with the worm, and as can be seen here, this is the specific antigens on the surface of the worm. So antibodies of IgE, they bind the worm with the eosinophils, and actually eosinophils, they express on the surface eosinophil receptors for the Fc part of or crystalloid fraction of IgE antibodies known as Fc epsilon R1. So this actually suggest that T helper 2 immune responses play an important role in immunity to schistosomes, as the T helper 2 produce interleukin number 4 that induces class switch to IgE antibodies. And the recruited eosinophils in infection with this parasite that causes schistosomiasis, actually these eosinophils, they produce eosinophil cationic protein and eosinophil derived neurotoxins, a major basic protein, as can be seen here, and these they have an anti-parasitic neutralizing or killing effect. And also neutrophils, they play a role in infection with schistosoma, and actually it plays this role in schistosomiasis japonica liver fibrosis following formation of granuloma. But however, in some instances, this parasite, I mean schistosomiasis parasite, has a capacity to evade effective immune responses, as it is highly motile. So through this motility, it can escape neutralizing immune responses. This is on one hand. On the other hand, the adult worm downregulates expression of surface antigens and cover itself by self-antigens, for example, glycolipid and glycoprotein, in order not to be recognized by the immune system. Thank you very much, Professor Ahmed Bolan.